All right. So good morning, everyone. Thank you all for joining us today, the New York State Center for Clean Water Technology seminar series, which happens every first Monday of the month. Today's topic of today's seminar is wastewater-based epidemiology. It's a joint presentation by Dr. Chris Gobler and myself, Arjun Venkatesan. And in today's seminar, we will be covering some brief introduction to the concept of wastewater-based epidemiology and also its application to track the prevalence of COVID-19 in communities, which is an ongoing work at the center. So I think it's a good starting point to the seminar is to understand a little bit about the perspectives on wastewater treatment plants. So wastewater treatment or sewage treatment in the US, it's an underappreciated infrastructure in the US essentially. And only recently people have started to realize the value and resources that it provides to us as a community. In the past, wastewater treatment was essentially considered as a decontamination facility and to meet standards and regulations. And the primary goal of wastewater treatment or sewage treatment was essentially to treat the sewage to an acceptable water quality so that it is safe enough to be discharged back into the environment. For those of you who are not very familiar with wastewater treatment or domestic sewage, it usually primarily a combination of human feces and urine, which is called as black water, and also contains gray water, which is essentially the water coming from washing, bathing, and meal preparation. So that's that's domestic wastewater. But in addition to that, we also get water and waste from various industries and businesses that's contained within the sewer shed or the wastewater treatment plant facility coverage area. Currently in the US, there are greater than 16,000 publicly owned wastewater treatment plants nationwide. But over the years, with research and advancement of technologies, people have realized that wastewater treatment plant can provide additional resources in addition to just treating your sewage. For example, the treated wastewater can be used for aquifer recharge. It is being reused for irrigation purposes. And in some cases with technology advancement, it's treated to such levels that treated wastewater can essentially be used for drinking water. The solids that's generated as byproducts during wastewater treatment also has values. It's rich in nutrients and other trace elements that it has been used as an agricultural fertilizer. It's more than 50% of the sludge produced in wastewater treatment plant is land applied for soil reclamation and agricultural fertilizers. In addition, energy is also generated during wastewater treatment processes. For example, biogas, biodiesel, and even solid fuels are generated from these sludge that's generated during wastewater treatment. And people have also realized that wastewater is rich in nutrients like phosphorus and nitrogen, which is of concern if released into the environment. But with advancement of technologies and research, we are now able to capture these phosphorus and nitrogen and recover them and use them for fertilizers as well. So due to all these benefits and resources provided by wastewater treatment plants, now the term wastewater treatment is being replaced as water reclamation facility or water resource recovery facility, given it provides so much of benefits and resources from protecting the environment to giving resources back to the community. So with that concept, like more recently, probably in the past decade, researchers have identified the chemistry of the raw sewage or the wastewater generated. It pretty much represents the community itself. So you could actually look for specific chemical and biological markers in sewage to get information about the community health and population health contained within the sewership. In other words, wastewater treatment plants can also be considered as a chemical observatory where you can study the behavior of the community itself. And this is where the concept of wastewater-based epidemiology first emerged. The concept was first introduced in 2004 by a scientist in Environmental Protection Agency. And essentially what the concept is we get exposed to chemicals or consume chemicals on a day-to-day basis, roughly about more than 1,000 chemicals we are getting exposed to on a day-to-day basis. These chemicals do not accumulate in the body, but however is metabolized and excreted, which enters the sewer lines and ultimately reaches the wastewater treatment plant. So one could essentially collect a sample at the wastewater treatment plant and look for certain biological or chemical markers to understand consumption of chemicals or exposures to chemicals that is happening in the community. So this is essentially the wastewater-based epidemiology concept. Once after the concept was introduced, it was first applied in European countries where they tracked the consumption rate of illicit drugs like cocaine and heroin in communities, where they were able to track the consumption trends in communities using wastewater-based epidemiology approach. The biggest advantage of wastewater-based epidemiology is that it provides near real-time information and accessible epidemiological data. These information is typically collected by surveys, by calling up random numbers and getting information about chemical consumption, which has its limitation, but doing this, it gives an unbiased and near real-time epidemiological data that could be used to assess public health. And another advantage is it is a non-invasive approach, meaning that it gives collective information instead of like giving individual persons information. So it is also helpful in collecting anonymous information about the entire community as a whole and public health information. So just to break down the concept again, for chemical consumption, after the drugs are consumed, the human body metabolizes the chemicals, and the metabolites are then excreted via urine or feces, which enters the sewage and is collectively collected at the wastewater treatment plant facility, where we could collect samples, representative samples, and analyze for these drugs. So where we could get the concentration of drugs in sewage. The concentration can then be converted to mass or load per day by multiplying with the flow of wastewater that can be measured at the wastewater treatment plant facility. And if the population of the community is known, this mass per day then can be converted to a consumption at milligrams per capita per day. So that's how the flow chart or the process of wastewater-based epidemiology works. So over the years, the concept, this approach has been applied for several markers. So they were first started with illicit drugs, which provided very robust data and trends in consumption patterns. Later, it was then extended to licit drugs like alcohol and nicotine consumption in communities. Now, more and more research are now focused on other pharmaceuticals and personal care products exposures in communities. This approach has also been utilized for looking at industrial chemicals like flame retardants or even PFAS. And it strongly has the potential to look at stress in communities, the dietary trends in communities by looking at specific markers. The wastewater-based epidemiology has also been applied for looking at population markers, where we can actually estimate the transient population that contributes to the sewage flow within the community. And more recently, and probably the most impactful application of wastewater-based epidemiology is looking at biologicals, essentially pathogens like disease-causing viruses and microorganisms that can inform on disease progress in communities. In the current seminar, we're going to have two case studies. The first case study is looking at the wastewater-based epidemiology application to look at alcohol and nicotine consumption in communities. And later, we're going to talk about how we could use this approach to track the prevalence of the ongoing pandemic, novel coronavirus disease, which Dr. Chris Gobler will be talking about as well. So moving on to the first case study, the wastewater-based epidemiology. This work was done in my previous lab at Arizona State University. So I'd like to acknowledge my colleagues, Dr. Cheng and Dr. Halden from ASU. So some background about how we are going about this wastewater-based epidemiology for alcohol and nicotine consumption. So let's first focus on alcohol consumption. After alcohol consumption, our body metabolizes alcohol and excretes them as ethyl sulfate and ethyl glucuronide. And the percentage of excretion relative to consumption is provided here as numbers. So what we are essentially doing is we are monitoring the metabolites and not as much as the alcohol or the parent chemical that we are consuming. So we chose ethyl sulfate as the metabolite to monitor for wastewater-based epidemiology because it was most stable in sewage and we were able to get strong signals in the sewage. For nicotine consumption, after nicotine consumption, we metabolize nicotine to form cotinine and also 3-hydroxycotinine. So for this purpose, for wastewater-based epidemiology application, we monitored cotinine and 3-hydroxycotinine as the metabolite of nicotine consumption to back calculate and estimate nicotine consumption in communities. Just to go over some of the calculations that we use to estimate the consumption rates so the CETS is the measured concentration of ethyl sulfate. It's one of the metabolite of alcohol, multiplied by the flow rate of the wastewater treatment plant. And CF is the correction factor, which I'll describe in a minute, divided by the population contained within the community and the density of ethanol. So this would then give us a result of milliliters of alcohol consumed per day per capita in the community. So the correction factor that we use is essentially to correct for the ethanol consumption. So we are monitoring only the metabolite. So we want to account for the factor or the fraction of ethyl sulfate released after the consumption of ethanol. And that is incorporated in our calculation. For nicotine consumption, so we use both cotinine and 3-hydroxycotinine as the metabolite to calculate the consumption rate. Similar to alcohol consumption, this is multiplied with the flow to get the mass per day, and then use the correction factor for the excretion patterns and divided by the population, which results in milligrams of nicotine consumed per day per capita in these communities. So this approach was applied to 3 US communities. 2 of these communities were located in Midwest region, and the 3rd community was located in the Northeast. These are relatively small communities with population from 45,000 to up to 125,000. The average daily flows of the wastewater treatment facilities serving this population range from about 8 million gallons per day to up to 28 million gallons per day. And the wastewater source is a little different for these communities. Like community 1 was dominated by domestic flows with little contribution of industrial wastewater. For community 2 and 3, there were more contribution from industrial and other sources as well. Community 1 and 2 have separate sewer systems, whereas community 3 is a combined sewer system with overflows from runoffs. So we collected 24-hour composited samples at the influent of these wastewater treatment plant facilities using autosamplers. And autosamplers was programmed to collect 100 to 150 milliliters of wastewater every 20 minutes for a period of 24 hours. So going to sample analysis, so we're looking at trace amounts of these nicotine metabolites. So we had to use some concentration step. For this purpose, we use solid phase extraction. So the typical sample preparation flow was we took 200 milliliters of wastewater samples. We added some isotopically labeled standards for QA/QC and recovery estimates, loaded onto SPE, eluted them, dried them, reconstituted in methanol, and analyzed using liquid chromatography and mass spectrometry. In case of alcohol, it was a lot easier because I think alcohol consumption was much higher, so we were able to detect them without any concentration step. So wastewater was centrifuged, and the supernatant was then diluted with water and directly injected into the instrument for detection of analytes. So let's look at the first data set for alcohol consumption trends. So the left panel here shows the actual concentration of the ethyl sulfate detected in the influent wastewater at these 3 different communities. As you can see, every single sample showed detection of ethyl sulfate, and the concentration varied over a period of about 2 years, close to 2 years. The concentration in community 1 seemed to be a little higher than in community 2 and 3. But when this concentration is then converted to milliliters per day per capita, is using the equations that was previously described, you can see now the pattern changes. So the consumption pattern between community 1 and community 2 were very similar. It was the consumption was ranged between up to 5 to up to 40 milliliters per day of alcohol per capita was consumed. But in case of community 3, we saw a lot more variance, and we saw like very high consumption rate during summer of 2015. And this was later we realized that community 3 actually has a national park within its sewage, which is kind of shut down during winter season, and it's open during spring and summer. And as you can see, the consumption of alcohol during summer increased. We believe this is because of the crowd that's coming to visit the national park during that time. So this also shows an importance of the calculations that you do for wastewater-based epidemiology. We need an accurate representation of population. Otherwise, your data could be biased in the estimates of consumption patterns. So moving on to nicotine consumption trends. Again, the left panel shows the actual concentration of cotinine and 3-hydroxycotinine, which are the metabolites of nicotine. It was consistently detected in all 3 communities. And when converted into consumption, which is milligrams per day of capita, again, we couldn't see any significant difference between consumption patterns. It's probably, this smoking is probably more consistent compared to alcohol consumption. And that's why we do not see any significant trends or differences between the 3 communities for nicotine consumption. Of course, we want to validate all these data, right? So we get this consumption pattern. The only way to validate is to compare our estimates with actual surveys that's done. So for this, we use the World Health Organization database, where sales statistics and interviews were used to get the consumption pattern for nicotine and alcohol. We also used National Institute of Alcohol Abuse and Alcoholism reports for state sales and shipment to get estimates on consumption patterns, and also CDC state system that uses a questionnaires by a random digit dialing system to select samples of adults with households or cellular phones to get information about their consumption of nicotine and alcohol. As you can see, the blue bar here is the average estimate that was calculated using the wastewater-based epidemiology approach. And the brown bars are the consumption that was reported by the state reports, and the dashed line is the US average consumption of alcohol and nicotine. As you can see, our results are very much in line with what has been reported by other traditional surveys. So this clearly shows the value of wastewater-based epidemiology, and if using the right approach, it could be as accurate in getting the information about consumption patterns of chemicals. The only outlier here is community 3, for which was much higher. Our estimates were higher compared to the state and US average. And this again relates back to the sewer shed itself, because the community 3 is a combined sewer flow, and also it has a national park. So the population numbers that we used for our calculation was incorrect, and it biased our results. And this clearly shows like potential of these, the wastewater-based epidemiology approach to look at chemical consumption. With that, I'll hand over the talk to Dr. Gobler, who will talk about the current work at the center.
Okay, thank you, Arjun, and thank you for a great introduction. Yeah, well, I think I'll start out by saying, obviously, it's been a very interesting, I guess it would be a year and a half. And it was about 1 year ago that we were all in lockdown, but yet Arjun and I still meeting. And you just saw the very strong background that Arjun has in wastewater epidemiology. And I think, and at some point Arjun can correct me, I can't remember if the first study had actually even been published on using wastewater epidemiology to study the COVID virus, or if there was just sort of internal talk of that. But regardless, the first successful studies of tracking the virus in wastewater were done and published out of Europe. And you may recall that Europe was just a month ahead of the US with regards to the first wave of the coronavirus. So anyway, it was last April we said, let's see if we can do this on Long Island. I think everybody had lots of different feelings about the sudden onset of the pandemic, and but one of the feelings we had is, wouldn't it be good if we can do something to help better understand the dynamics of this pandemic locally. So next slide there, Arjun. Okay, so just for perspective, we all know where we are in the pandemic, and here are the as you can see, the first wave way back in the beginning of 2020. That first bump there with the peak is somewhere around April, and a slow tail thereafter. Then the second, maybe probably shouldn't call it wave, but I'm just going to use that for no better adjective at this point in time. Last summer, and then the very large wave that was the onset that coincided with Thanksgiving, Christmas, and New Year's. And there are some people looking at the most recent data and the fact that the levels now are higher than they were last summer, despite there being actually fewer testing now than there was last summer. People are calling this a potential fourth wave. Next slide, Arjun. So, why is it that the coronavirus has been so difficult? I think we all know this, but I'm giving this information because it provides important context for why we're doing what we're doing. So people are most apt to spread the virus before they know they are sick. So it's pre-symptomatic spread of the virus that is so troubling. People don't realize they have the virus and are therefore passing it on to people, and then finding out 2 days later, oh, I'm sick. But it was the past 48 hours that have probably been some of the most dangerous. And then, of course, we also know this is an asymptomatic virus. And so that is also a problem. So you have people spreading the virus. They don't know that they're sick. They don't know they're carrying the virus. And so that is one of the reasons that this approach potentially has such great value. So I think next slide, Arjun. Okay, so this is a little detail. This comes out of a Nature paper that was published last year. And essentially, if you look at it, I'm going to start from the top and work to the bottom. So if we work with the very first horizontal line there, it's essentially supposed to show that the first blue horizontal line is upon infection, and then there's going to be a certain time after which someone's first been infected, at which they know they're infected, and that's either they have a rapid diagnostic test, which is the second blue line vertically, or the third vertical blue line, which is if they have a delay in that testing. And you may recall that, for example, last summer, there were very significant delays in testing. But when it comes to the virus, if we look at then the next horizontal line where it says viral shedding into wastewater, we know that people start what they call shedding the virus in their feces shortly after infection, and certainly before they display any symptoms. So that means that people are shedding this virus when or before they're actually able to spread the virus. So right there, that tells you the power of using this as a diagnostic as opposed to using how people are feeling. And so if we go to the third line, that's a study that was done specifically looking at a sewage treatment plant, but using sludge. So not the wastewater influent, but the sludge settled shortly after the influent. And so that was a very good study. Again, it was one of the first studies out of Europe, and it showed for certain that the virus is showing up in the sludge. But as it turned out, the sludge sampling, because of the time course between the influent and it settling out, I think also maybe processing the samples, it offered no advanced lead time over rapid diagnostic testing. It would have an advantage if there was a delay in such testing. But then if we go to the final line, this is the value of sampling the influent, and the idea that you can actually have an up to 1 week advance notice of an uptick in infections by monitoring the influent, because by doing that, you're actually detecting the virus in people, firstly, who of course are asymptomatic, but also people who are shedding the virus and don't even know that they're infected. And in either case, those people are spreading the virus. Okay, next slide, Arjun. Okay, this is just to show you that what we're doing is not novel. There are, as you can see, across the planet Earth, 235 universities engaged in a similar study in over 50 countries. You can see the very strong clustering of the approach in Europe, somewhat in the Middle East, and obviously in the United States. Next slide, Arjun. And of course, in the United States, you can see it's a very popular approach in the Northeast and, frankly, the eastern third of the U.S., less so to the West, but that's just tracking really the presence of universities by and large, at least. And then you can see in the Northeast, when you use a little bit smaller symbols, you see that, well, there's not too many necessarily universities doing it around here, a few, 3 in New York City, but we're certainly on the map. This is, again, a global dashboard, and we've been acknowledged as one of the universities engaged in studying the virus. Next slide. Okay, and then some of these are of these individual universities doing these studies have very advanced diagnostics, and so I'm very fond of Yale University's tracking. They're tracking, I believe, 8 sewage treatment plants across Connecticut. It turns out, it's very clear when you look at their data that I think they had already agreements for doing some work with these sewage treatment plants before the onset of the pandemic. But here's just something I pulled off of their website, where they're putting up data in the upper graph, the caseload, the virus, detection of the virus within the wastewater, and then below the number of cases in a given watershed. And for some reason, they recently have not been updating as much as they had been, but up through, I believe, February or so, it was tracking quite nicely. And I guess the one thing I will point out that this data set shows is that, as you saw in that theoretical figure, you're supposed to get a lead time with regards to the virus and the onset of waves. And you see here, and you focus specifically on what was going on in the fall, October. You need to really squint your eyes to see that the uptick in the viral copies in the wastewater came before the uptick in number of cases in the population. And of course, it's so important to emphasize and really get an appreciation for this. Is like, despite the fact that now, this is, you could say, commonplace. There's hundreds of universities doing this. Obviously, this is a field that is 1 year old. And so everybody is learning on the fly. Many papers coming out at a rapid pace, but frankly, there's a lot to learn, and frankly, that's also what makes it quite an exciting course of study. Next slide. Okay, so our approach here. So in all cases, for all sites, I'm going to describe the different sites that we're studying. The samples that we're getting are 24-hour composite samples, and so the idea there being that you're not just getting a single point in time, which could be influenced, say, if you collected the water at a certain time of day, maybe there's a bigger signal from a bigger fecal signal than there would be at another time of day. And so a sort of a standard practice for wastewater epidemiology in general, and certainly for monitoring the virus, has been getting these 24-hour composite samples where a small volume of water is pulled every 15 minutes or so. And then what you actually analyze is the composite of all of those samples collected over 24 hours. So our first samples were collected again just a year ago, where very thankfully the associate director of the New York State Center for Clean Water Technology, Frank Russo, in his former career as a wastewater engineer, actually helped design the Riverhead and Patchogue sewage treatment plants. And he was able to connect us with the people operating those plants to begin sampling those all the way back last April. Next line there, Arjun. And then we began discussing that. We very thankfully have regular meetings with Suffolk County and with the DEC, and Suffolk County, we presented to them, I believe, in our quarterly meeting last spring, what we were up to on this front, and they were quite interested in getting information. And then they facilitated, thanks to actually efforts by Arjun, getting samples from the Bergen Point sewage treatment plant. I'll show a map of it later, but it happens to be the largest sewage treatment plant in Suffolk County. And following Arjun's advice on sort of standard protocols for wastewater epidemiology, we began all sites by collecting samples once a day for a week, weekly for 2 months, and then slowing down to monthly thereafter. I'll emphasize, in the beginning, we froze the samples we collected. We were very eager to get going, but our eagerness was ahead of our ability to actually do the work. Frankly, we had the idea, but no assays, and frankly, not even a lab that was approved for doing this work. And so our first go around was the collection of the samples, and those samples being immediately frozen in a -80 freezer. Next line, Arjun. This fall, we had an exciting, I would call, mini project at the university, where we began tracking the effluent out of the dorms on campus to look for the signs of the virus in the wastewater effluent there. And that's when we transitioned actually to analyzing the samples immediately. So not freezing them, but collecting them and then processing them immediately upon collection. Next line. And then this fall, we continued discussions with Suffolk County. We updated them on our progress with regards to both the campus study and the other studies. And they became very interested in applying this in a very regular basis and getting rapid data out of their largest sewage treatment plant. And particularly this, I guess, became even more of an issue as the second wave in New York became clear was happening as of the fall of 2020. Next slide, Arjun. Okay, so just some quick pictures. This is for our just our campus study, where we had to actually build our own composite samplers, and I'd like to acknowledge my research technician, Craig Shanone. We've got information on how this would be built, and Craig, being very handy, got all of the parts. This is just simply, the sampler is the box on the left, and you can see next to that is a manhole cover, and that cover open. This is on campus. And I'll mention that what we were able to do is that there's a single sewer line or effluent line that goes from 80% of the dorms towards the on-campus sewage treatment plant. And the university buildings and grounds people were very cooperative in helping us access this site, fenced-in site, so we could collect that effluent from the dorms literally on its way to the sewage treatment plant. Next slide. And this just shows you the inside of our composite sampler. You can see it's insulated. The sample on the lower right, it's on ice, so we want to keep the sample as cold as possible to prevent degradation. We're working with an RNA-based virus that's frankly not very stable on its own, particularly in wastewater, so we need to take that precautions. The upper right is our peristaltic pump that collects the sample out of the sewer hole there, and the whole thing's driven by a battery. Next slide. And this is what our samples look like. Not too bad considering what they're coming from. And we would always try to collect duplicate samples. Next slide, Arjun. And all of our work was done in a level 2 biosafety lab. This actually required approval from the university. They had to approve actually everything. In the very beginning, I just, again, it's important to not lose sight. I'm very used to just doing science and sort of pro forma and let's go. In this case, when I first approached the university about doing this, remember it was early April, as deaths had not even been peaking yet in New York. And frankly, people didn't even know how contagious the virus might be from wastewater. So we had to set up a whole separate brand new lab from scratch with a whole new biosafety cabinet and all sorts of protocols about donning and doffing PPE before working with these sorts of things. But anyway, it came together not as fast as we'd like, but we now have this facility and it's working quite well for us. Then this I just mentioned is in my lab in Southampton. There's a few ways to get at the virus or get the virus out of the wastewater. In the beginning, we tried a centrifugation approach. Turns out, as everyone probably knows, with the pandemic, there's been global shortages on many things. Maybe you've heard of this, maybe you've experienced this yourself. There were global shortages on scientific supplies. And so our first go round, what had been the standard protocol, was using a centrifugation step to concentrate the virus. Turns out those supplies were quite, just were not available. Everything was backordered. And I credit my postdoc, Dr. Deepak Nanjappa, who looked into the idea of using a polyethylene glycol approach for concentrating the virus and settling the virus out. Turns out this has worked very well in our hands for getting good recoveries of the virus and actually save some time. So that's what we're doing now is we're concentrating the virus and then centrifuging it as opposed to buying individual centrifugation columns. Next slide. And then the approach we're using for detecting the virus is something we call digital PCR, also known as third generation PCR. So PCR is polymerase chain reaction, and in its first generation, the idea was to find a given DNA sequence in a sample. You could then create a complement DNA strand with potentially a fluorescent probe to anneal to the sequence that you're interested in and give you a signal. And so in the first generation, it's really a yes or no question. And then came quantitative PCR, in which case you'd successively go through multiple PCR amplification cycles. And in doing that, and by having a standard curve, you can actually not get a yes or no answer, but get an answer with regards to how much of that gene of interest or sequence of interest is in your sample. But there's a lot of caveats with that approach. There's issues with interference. You have to have standards. And so then what they call the third generation here is digital PCR, where by you're conducting individual PCR reactions like the first generation, but you're actually doing about 20,000 of those on a single chip. So you get 20,000 yes or no answers. And based on that, you actually come up with a concentration. Next slide, Arjun. So if you look in the upper left there, once you've concentrated and extracted your RNA, you're then reacting that with a set of primers and probes. And then you can see in the hand, the gloved hand there, the size of the chip, right? So this is like almost the size of a thumbnail, but yet in that thumbnail sized chip, there are 20,000 reaction wells in which you're reacting your sample with the primers and probes. You seal up that chip, put it into the machine, and then you're ready to do your PCR next step. And I'll mention this again later, but in addition, there's multiple targets for PCR that we're using here. We have two different genes from the from the virus that we're looking at, the N1 and N2 genes. We have a human gene, RNAsP, and then another coronavirus that's from actually a vaccine that we use as a positive control. I'll go through this more later, but you can see there in the cartoons the idea that if you've got the right match, when you have the annealing of the primers to your target DNA, it then yields a fluorescent probe, and that's what you're actually detecting. It's the fluorescent dye that gets knocked off of your probe. Next slide. So again, it gets quantified within the machine. The chip is inserted, and then you're just looking at the number of positive signals versus positive fluorescent signals versus those that do not fluoresce. Next slide, Arjun. And you can see that here in sort of graphical form. The tame and Vic are two different fluorescent dyes. And so what you can see here is that you can see there are many non-positive wells, all the yellow ones, and then a handful that react with the FAM dye, and those are the ones where you know you had a copy of your target gene sequence. Next slide. Okay. So again, there's two nucleocapsid genes from the virus that we're using to track the virus within wastewater. I should mention the CDC put out specific protocols for wastewater testing of the virus. There were sort of derivatives of the initial testing that was put together for humans, and frankly, to see if people had the virus. That original protocol was based on quantitative PCR. It was based on, as I mentioned before, using centrifugation only step for concentrating the virus. So we sort of have tweaked it to optimize results and to maximize recovery. But with all our reactions, we have a positive control, a negative control. We have this calf coronavirus vaccine that we're using as an internal standard, where we add it to samples before we concentrate them, before they extract them, to see how much we get back out of the concentration and recovery step. We also have an additional gene we're looking at called the RNAsP gene that theoretically should be a marker for just of the number of humans within a watershed, because everybody should be shedding that gene at an equal rate as well. And then we're further then trying to normalize our data to different chemical parameters within the wastewater, things like ammonium, biological oxygen demand, and I think Arjun has some chemicals that are of interest as well. So just moving on to some results. So here is what things have looked like in Suffolk County. We were thankfully spared the second wave in the summer. You recall we saw I showed you earlier what the national caseload looks like. But you can see that we all are familiar with what happened last spring, and I guess probably all familiar with all of this. So we, just with regards to what we did, we sampled Riverhead and Patchogue for the first wave. Bergen Point began in June, and we were on campus at Stony Brook in the fall. And I'll just show some taste of that data now. Go ahead, Arjun. So here's our very first data set. Frankly, before we had optimized our methods, but still, even in doing so last spring, we were detecting the virus, and that we saw a decay of the virus in the wastewater that paralleled the decay in caseloads in the watersheds. You can see we had high levels when, frankly, the pandemic was peaking and caseloads were peaking in Suffolk County, and as things tailed off, we then went to undetectable levels. This was using quantitative PCR, which in our hands has been a little bit less sensitive than a digital PCR. So the zeros that we had in May, were we to go back and redo those with digital PCR, I believe we'd have some detections there as well. Next slide, Arjun. So to give you a taste of what things looked like on campus at Stony Brook this fall, we got kind of a late start, but we did get some good data. So Arjun, let's look at the thankful news that despite there were cases on campus at Stony Brook, to our knowledge, as this was the case certainly as December, and I think it's been since that, we have not had university employees or students be hospitalized or any fatalities. So that's been at least a happy part of this and something to consider as I show you cases, at least on campus. So this plot is from the fall semester, and what it shows in the blue bars is the testing of people on campus. And in the orange line is the number of the percent positivity rate as a 7-day rolling average on campus. And as you look at these numbers, firstly, recognize there the university was ramping up to testing more than 1,000 people each day during the weekday on campus. And while there were some positives, note that these positivity rates are very low, right? So we peaked in December at 0.6%. So the university had a lot of different safeguards in place, and clearly they were useful. I will just, as an anecdote, point out that very first high point in September was that's December. So up in September, all the way back, the very first high point when the semester began, the orange high point there was almost 20 cases in a single day, and I thought we were headed towards closing down. But you can see things got very quiet thereafter. In fact, we had no cases reported for quite an extended period of time in September and October. But then things started picking up again. Next slide, Arjun. This slide is a little bit busy, but just to break it down, we only started sampling at the very, and really, frankly, end of October and into November. And when we first started sampling, we weren't getting detections. Remember, this is the effluent coming out of the dorms. But we started getting hits for both of the genes in November, got very high levels, as you can see, by the beginning of November. And then thereafter, we continued to have detections. So there was positive levels there, but not as high. And as you may recall, another caveat in all this is that well, maybe people don't know this, the dorms were closed starting Thanksgiving. So we went to all remote. So there were in-person teaching. I should qualify that with that fact. In fact, I taught some class in the fall in person. But by by Thanksgiving, everything was virtual. So clearly there was virus in the wastewater coming out of the dorms by November. So in many ways, you might say that things might have shut down just in time. So that original plan to shut things down by Thanksgiving, in hindsight, was a very good one. Next slide. Okay, and this shows the caseload and that same set of data. And so what it shows is that there were some early cases when we were getting only very low detections. And it was only after the cases built up that we started getting detection in the effluent. Next slide, Arjun. And this just showing, again, the percent positivity rate. And again, just similar idea that we were picking things up as the caseloads increased on campus, but not sampling as frequently as probably we would have liked, and not probably enough maybe before samples as well. So this brings us to the next and final data set, I believe. Next slide, where we've been collaborating with the county to sample from the Bergen Point sewer treatment plant, which encompasses that southwest sewer district. So that big area in red in the southwest corner of Suffolk County. As I said, that by far the biggest sewer district in all of Suffolk County. I believe I don't have the precise number on the tip of my tongue, but hundreds of thousands of people that contribute to this sewer shed. Next slide, Arjun. And again, we can remind ourselves of what the caseload looked like. This is for the entire pandemic. And if you go to the next slide, Arjun, I lopped off the beginning here just to see, this is when we were actually sampling. So we began sampling at Bergen Point in June when caseloads were quite low. And then you can see, we all see what happened in the fall and where we are today. Next slide. So this is the N1 gene in the influent at the wastewater treatment plant. And you can see actually the patterns here line up not too bad. In general, what you're looking at is the actual individual measurements and then a rolling average. I believe this is a several week rolling average And so, you see in the very beginning when we started, I think it was the first week of June, levels were low. We did have one sample that was high and a couple of other high ones thereafter, but in general, things were quiet, relatively so, through the summer and into the early fall. And then you see in the fall, things start picking up. And then, fairly high levels with certainly some variance. And that's one thing that we're learning. There's definitely variability in this data. And that's both analytical variability and also even samples collected side by side. And again, this being a new field, there's a lot of unknowns. And if you go to the next slide, Arjun. But now this is just looking at the caseloads in Suffolk County, and then looking at above and below the N1 gene copies per liter in the influent. And so again, plenty of variance, but what is of interest is that you can see we actually had some samples in the fall in November that were ticking up just before things in the caseload started jumping off in Suffolk County. So giving us the hint that perhaps, monitoring this virus, this was done in retrospect. We didn't get to do the analysis of the fall samples, frankly, until the winter. But with the support from the county, we were able to do that. And what it shows is, like I said, there did seem to be some hints of the caseloads or the virus becoming more prevalent before the caseloads got high. And certainly we're at elevated levels now, but with plenty of variance. Next slide, Arjun. And this is just to show that we have also been looking at that second coronavirus nucleocapsid gene, and in our hands, and frankly, it's been shown globally, it just doesn't track as well. I think it's a lot less stable than the other gene, and therefore the results, there's maybe some hints of some, obviously high levels in all of our fresh samples. That's the other thing, that the ones in January on were ones that were analyzed fresh, but in the frozen ones, not a lot going on. But frankly, this is the global case. And if you look at the papers being published, people are really focused on the N1 gene and not the N2. Next slide. Okay, so I guess this is what we had to conclude on. I think Arjun maybe will give some final statements after this, but again, there was the promise of detecting infections from people who are contagious but not yet sick, and there's some examples that may have been the case in the fall. Next line there. And again, to detect waves in the community, and so certainly we're seeing that now, and maybe saw that in the fall. And so just on the Riverhead front, we did pick up the virus there at the peak of infection. And similarly, we did, that should say Bergen Point STP, not STOP. But we're definitely seeing that we were picking up the virus and paralleling caseloads, both on campus and at Bergen Point. And so, very clearly, I think, no matter what comes of our study, clearly this is a global approach now. And I think going forward, this approach is going to be likely implemented in a permanent way in some places for looking at diseases in communities. And I think now, Arjun, you're going to wrap up here.
Yeah. So as we all heard today in the seminar, like wastewater-based epidemiology certainly have a lot of potential looking at public health and looking at hidden outbreaks of diseases. But there are certainly challenges and uncertainty in the data that we collect from wastewater analysis. And I want to go through some of those points or places where we need to have some consideration on how we sample. So especially sample collection can really impact the data we collect. For example, composite sample is what is more important for wastewater-based epidemiology approach versus grab samples. In grab samples, you don't really get complete understanding of what's happening over 24 hours. So very high frequency sampling, like every 10 minutes or even less, or maybe continuous sampling throughout the 24-hour period would be more applicable for wastewater-based epidemiology and will give it more robust data. Sample storage and handling, as Dr. Gobler mentioned, it really impacts the analysis of RNA virus, like freeze-thaw cycles can impact detections of the RNA that we're looking for. Sampling location can also impact the data that we observe. Like typically we have access to the wastewater treatment plant facility. It's easy to collect influent of wastewater, but moving closer to the source would give you more accurate results. Like people are now collecting samples at different sewer points, pipeline points to get more data or to understand a subset of the sewer shed progress in coronavirus and other wastewater-based epidemiology applications. Fate of chemicals and biological markers in sewage is also something that you need to consider. Many of these chemicals and biological markers are not stable in sewage. Typical time that the sewage travels from the source to the collection point at the wastewater treatment plant could be up to 8 hours. So in 8 hours, you could have some degradation of the signal that we are looking for. So we need to consider the half-life or fate of these markers when we do some calculations. Of course, analytical methods can also vary. Sewage is a very complex matrix and it can impact the performance of the analytical methods that we employ for the detection of these markers. There are also uncertainty in calculations, and this was highlighted in the alcohol and nicotine study in this seminar, where an incorrect use of population data can really bias the consumption rates or exposure rates that we determine using wastewater-based epidemiology. Also, we need to account for dilutions happening while sampling. So we need like more normalizing parameters to account for those dilutions in sewage. Population, again, as I highlighted, is important to know. If you use the population of the sewer shed itself, it's going to bias your data because there could be transient population, especially this is applicable for New York City, where a lot of neighboring states, people are coming into New York City for work, and they could bias the flow of wastewater and also the levels of markers that we're looking for. So these transient population need to be captured when you do the calculations to estimate exposures and consumption. With that, of course, we need to acknowledge a lot of people behind this research. Drs. Deepak Nanjappa, Mian Wang, and Chengchuan Li, who are leading these wastewater analysis for Suffolk County samples. Also Anne and Hannah to help with sampling and analysis. Dr. Mao who collaborated with us for this project. The CCWT team and Gobler Lab. I also want to acknowledge Dr. Jing Shen and Rolf Halden from Arizona State University, where for the study conducted on alcohol and nicotine. Thanks to Stony Brook University, New York State, Suffolk County, Bloomberg Foundation, and Roche Foundation for support for some of the work that we did. And thank you all for your attention, and we'll be happy to answer any questions you may have.
And I see, I'm going to start during the questions because I see Karahan had asked a question, legislator Karahan. And she specifically was asking about testing for different variants, the UK variant, etc. And so to our knowledge, the N1 gene is conserved across the variants. So we are not able with our approach to discriminate amongst the variances. But we are the variants, but we are able to pick up the different, if they're there, we're not missing them. Jim Meyer asked if there was any evidence of transmission of COVID via wastewater, and we believe the answer there is no. So there was a big conference, global conference held on this subject. I want to say it was last summer. And I think that was sort of the consensus statement out of that, that even though we're detecting the RNA of the virus, we are, that it's non-infective. So that's the current consensus on the topic. Another question here from John Cornridge. And he's saying, wondering, for public health or epidemiological purposes, is the inclusion of a sampling station part of the current design standards? That's a good question. And I don't know. For design standards, that's a good question. I wonder if Frank would on. Frank, if you're there, I don't know if you heard this question, but you might be the best one to answer it. And that is, for current design, I think the question is, if when designing sewage treatment plants, you have to include an influent sampling station as part of the design.
The permit by which the plant operates will specify the influent point and the sample location for the effluent. So yes, by design, we have to include that.
Okay, good question, and the right person to give the answer. Thank you, Frank, and thank you for your question, Jonathan. Additional questions, you can put them in the chat, or I think if you wanted to unmute, that would be okay too.
So I'm unmuted. This is Bruce, because I type slowly. Anyway, thanks a lot. I may have missed it because I missed the very beginning of the talk. Did you guys get a chance to do much to follow up on Jim Meyer's question about transmission vector from wastewater? We had some really, I thought there was some irresponsible news out of Scripps in the spring about not walking on the beach because there was sewage discharged into the ocean. And if anybody was going to get exposed, I would have thought it would have been wastewater treatment workers, you know, where there's aerosolization at the plant. But anyways, did you get a chance to do any effluent testing where you could look at removal efficiencies in any of the plants?
Yes, and in fact, I wouldn't make this my final answer, but in our initial assessment on the effluent, we were not seeing any virus, so that it was being graded away before it left the plant. Which would, yeah, I agree, Bruce, that scary story that was put out by someone in Scripps was totally off base and part of, well, the kind of
just a little bit of dilution in the Pacific Ocean, and there's been a fair bit of work, some work on SARS that suggested decent wastewater removal.
Yeah, that's basically what we had seen as well. Nils, I see you have your hand up.
Yes, thank you very much. Related to Karahan's question, I mean, there could be a great opportunity here, right, to figure out what variants are out there in the community, since you have these composited samples, even though the gene that you're using right now does not allow you to do so. Do you know if this is tried somewhere else, or because that will probably be something that will be with us for maybe even years to come, right?
Yeah, and I think that certainly, I mean, that I can't pretend to know a ton about developing the level at which these variants are different, but theoretically, if there are genomic differences, then primers could be created for these different variants and detect those different variants, and so you could then look at the total amount of virus, and then in addition, the relative contribution by the different variants. But to mine, and I bet you there are people working on that right now. But that's, yeah.
Thanks.
Any other questions? Well, hearing none, on behalf of Arjun and myself and the center, I thank everyone for their kind attention and their interest in the topic, and we will be looking forward to giving our next public seminar first Monday in May. So thank you everyone for tuning in. And
thank you for all your work, Chris.
Oh, Thank you.
And Arjun. Thank you, everyone.
Thank you. And I wish everyone a good week.